let's continue with the previous session. So in this video we are going to learn about some other topics related to viral and bacterial diseases. So their causative organism, mechanism, symptoms, etc. So first, influenza, caused by the influenza virus, transmitted through respiratory droplets. Symptoms include fever, cough, sore throat, muscle aches and fatigue. Treatment includes antiviral drugs and rest. Then COVID-19, caused by the SARS-CoV-2 virus, transmitted through respiratory droplets. Symptoms include fever, cough, shortness of breath, loss of taste or smell and body pain. Treatment includes supportive care, antiviral drugs and vaccines. Then tuberculosis, caused by the bacterium Mycobacterium tuberculosis, transmitted through respiratory droplets. Symptoms include cough, fever, night sweats and weight loss. Treatment include antibiotics. Then malaria, caused by the parasite Plasmodium, transmitted through mosquito bites. Symptoms include fever, chills, headache and muscle pain. Treatment include antimalarial drugs. Then hepatitis B, caused by the hepatitis B virus. The hepatitis B virus, transmitted through infected body fluids. Symptoms include fever, fatigue, nausea and jaundice. Treatment include antiviral drugs and vaccination. Then Lyme disease, I'm not sure about the pronunciation, caused by the bacterium Borrelia burgdorferi, transmitted through tick bites. Then symptoms include rashes, fever, headache and joint pain. Treatment include antibiotics. Then cholera, caused by the bacterium Vibrio cholerae, transmitted through contaminated water or food. Symptoms include diarrhea, vomiting and dehydration. Treatment include rehydration therapy and antibiotics. Then HIV or AIDS, caused by the human immunodeficiency virus, transmitted through infected bodily fluids. Symptoms include fever, weight loss, fatigue and opportunistic infections. Treatment include antiretroviral therapy and prevention measures. Then streptococcal infections, caused by the bacterium Streptococcus, transmitted through contact with infected individuals or surfaces. Then symptoms depend on the type of infection but can include sore throat, skin infections and pneumonia. Treatment includes antibiotics. It is important to note that the treatment of viral and bacterial diseases can vary depending on the severity of illness and the individual patient's needs. In addition, prevention measures such as vaccination, good hygiene practices and avoiding contact with infected individuals can also help reduce the spread of these diseases. Then biomolecules, bonding and stereochemistry. Biomolecules are organic molecules that are essential to life, including carbohydrates, lipids, proteins and nucleic acids. These molecules are held together by various types of bonding and exhibit stereochemistry that is critical to their biological function. Then bonding, covalent bonding. Covalent bonding is the most common type of bonding in biomolecules. It occurs when two atoms share a pair of electrons, creating a strong bond. Examples of covalent bonds in biomolecules include the peptide bonds that link amino acids in proteins and the phosphodiester bonds that link nucleotides in nucleic acids. Then ionic bonding. Ionic bonding occurs when an atom donates an electron to another atom, creating positively and negatively charged ions that attract each other. An example of ionic bonding in biomolecules is the bonding of enzymes to their substrates. Then hydrogen bonding. Hydrogen bonding occurs when a hydrogen atom covalently bonded to an electronegative atom such as nitrogen or oxygen is attracted to another electronegative atom. Hydrogen bonds are relatively weak, but they are important in stabilizing the structures of biomolecules such as the double helix structures of DNA and the secondary and tertiary structures of proteins. Then stereochemistry. Stereochemistry is the study of the three-dimensional structure of molecules and how it affects their biological function. Biomolecules exhibit stereochemistry due to the presence of chiral centers, which are carbon atom bonded to 4 different groups. Chiral molecules have 2 enantiomers or mirror image forms that have different biological activity. For example, the amino acid alanine has a chiral center and 2 enantiomers, L-alanine and D-alanine. L-alanine is commonly found in proteins and is biologically active, whereas D-alanine is not commonly found in proteins and is not biologically active. Then stereochemistry is also important in the interaction between biomolecules, such as the binding of enzymes to their substrates. Enzymes are specific to certain enantiomers of substrates, and the wrong enantiomer may not fit properly and therefore not be processed correctly. In conclusion, bonding and stereochemistry are important aspects of biomolecules that determine their function and interactions in biological systems. Understanding these principles is essential for understanding the behavior and properties of biomolecules and their role in biological processes. Then Ramachandran plot in detail. The Ramachandran plot is a graphical representation of the allowed regions of phi and psi angles in a polypeptide chain, allowing the conformational preferences of amino acids in proteins. It was first proposed by G. N. Ramachandran in 1963 and has become a widely used tool in the analysis of protein structures. The Ramachandran plot is divided into 4 quadrants, each corresponding to a different secondary structure: alpha helix, beta sheet, polypropylene 2 helix, and random coil. The angles phi, the angle between the nitrogen and alpha carbon atoms, and phi, the angle between the alpha carbon and carbonyl carbon atoms, are plotted on the x and y axis respectively. The major points of the Ramachandran plot are as follows. Allowed regions. The majority of amino acids fall into the allowed regions of the plot, which correspond to the most energetically favorable conformations of the polypeptide chain. Then disallowed regions. Some regions of the plot are disallowed, which correspond to sterically unfavorable conformations of the polypeptide chain. Then outliers. A few amino acids may fall outside of the allowed regions due to unused conformations or interactions with neighboring residues. The steps involved in constructing a Ramachandran plot are: obtain the coordinates of the protein structure, usually from a crystallographic or NMR study. Then calculate the phi and psi angles for each residue in the protein using standard methods. Plot the phi and psi angles for each residue on the Ramachandran plot. Analyze the distribution of points on the plot to determine the secondary structure content and identify any outliers. The Ramachandran plot is a useful tool for studying protein structures because it allows researchers to visualize the distribution of secondary structure elements and identify unusual conformations. It is often used to validate the quality of protein structures determined by X-ray crystallography or NMR spectroscopy. In addition, the plot can be used to design new proteins or predict the effects of mutations on protein stability and function. Next, the Ramachandran plot is a useful tool for studying protein structures because it allows researchers to visualize the distribution of secondary structure elements and identify unusual conformations. It is often used to validate the quality of protein structures determined by X-ray crystallography or NMR spectroscopy. In addition, the plot can be used to design new proteins or predict the effect of mutations on protein stability and function. Then glycolysis regulation. Glycolysis is a metabolic pathway that breaks down glucose into pyruvate. The regulation of glycolysis is important to maintain energy homeostasis in the cell. Here are the details of regulation of glycolysis. Feedback inhibition. One of the most important regulatory mechanisms of glycolysis is feedback inhibition. In this mechanism, the end product of glycolysis, ATP, binds to the enzyme phosphofructokinase 1, PFK1, which catalyzes the third step of glycolysis, inhibiting the enzyme and the entire pathway. This ensures that glycolysis does not continue if there is sufficient ATP in the cell. Allosteric regulation. Another mechanism of regulation is allosteric regulation. In this mechanism, certain molecules can bind to the enzymes involved in glycolysis, changing their conformation and activity. For example, the molecule fructose 2,6-biphosphate activates PFK1, stimulating glycolysis. On the other hand, citrate, an intermediate in the citric acid cycle, inhibits PFK1, slowing down glycolysis. Then hormonal regulation. Hormones can also regulate glycolysis. The hormone insulin stimulates glycolysis by promoting the uptake of glucose into cells and by activating the enzymes involved in glycolysis. The hormone glucagon on the other hand, inhibits glycolysis by decreasing the concentration of fructose 2,6-biphosphate. Then phosphorylation. Phosphorylation is another regulatory mechanism that can affect glycolysis. When the cell has high energy demand, such as during exercise, glycolysis is stimulated by the phosphorylation of PFK2 FBPase2. This enzyme converts fructose 6-phosphate into fructose 2,6-biphosphate, which stimulate PFK1, thus activating glycolysis. Then gene expression. The expression of the genes that encode the enzymes involved in glycolysis can also be regulated. For example, the transcription factor hypoxia-inducible factor HIF1 can upregulate the expression of genes involved in glycolysis in response to low oxygen levels. The regulation of glycolysis is important because it helps to maintain energy homeostasis in the cell. The pathway must be regulated to prevent unnecessary consumption of glucose, which is an important energy source for the cell. By regulating glycolysis, the cell can respond to changes in energy demands and maintain energy homeostasis. Then Krebs cycle. The Krebs cycle, also known as the citric acid cycle or TCA cycle, is a central metabolic pathway in aerobic organisms. It takes place in the mitochondria of eukaryotic cells and cytoplasm of prokaryotic cells, and it is responsible for generating energy-rich molecules such as ATP, NADH, and FADH2. The cycle is composed of a series of enzyme-catalyzed reactions that convert acetyl-CoA into carbon dioxide, producing energy in the form of electron carriers. The Krebs cycle can be divided into 3 stages. The first stage is the conversion of acetyl-CoA to citrate. The second stage is the conversion of citrate to isocitrate, and the third stage is the oxidation of isocitrate to alpha-ketoglutarate, which enters the next stage of the cycle. The cycle also involves the regeneration of oxaloacetate, which is used in the first stage to combine with acetyl-CoA to form citrate. The major steps of the Krebs cycle are citrate synthetase. The first step is the conversion of acetyl-CoA and oxaloacetate to citrate by the enzyme citrate synthetase. This step is irreversible and is the rate-limiting step of the cycle. Then aconitase. Citrate is then isomerized into isocitrate by the enzyme aconitase. Then isocitrate dehydrogenase. Isocitrate is oxidized to alpha-ketoglutarate by the enzyme isocitrate dehydrogenase, producing NADH in the process. Then alpha-ketoglutarate dehydrogenase. Alpha-ketoglutarate is then converted to succinyl-CoA by the enzyme alpha-ketoglutarate dehydrogenase, producing NADH and carbon dioxide. Then succinyl-CoA synthetase. Succinyl-CoA is converted to succinate by the enzyme succinyl-CoA synthetase, producing GTP, which is later converted to ATP. Then succinate dehydrogenase. Succinate is then organized to fumarate by the enzyme succinate dehydrogenase, producing FADH2 in the process. Then fumarase. Fumarate is then converted to malate by the enzyme fumarase. Then malate dehydrogenase. Malate is oxidized to oxaloacetate by the enzyme malate dehydrogenase, producing NADH in the process. The Krebs cycle plays a crucial role in cellular respiration, as it is responsible for generating ATP, which is used as an energy source by the cell. It also produces electron carriers such as NADH and FADH2, which are used in oxidative phosphorylation to produce additional ATP. The Krebs cycle is regulated by several mechanisms, including allosteric regulation, substrate availability, and feedback inhibition. Allosteric regulation allows the binding of a molecule to an enzyme at a site other than the active site, which can either activate or inhibit the enzyme. For example, citrate can bind to and inhibit citrate synthetase, while ADP can bind to and activate isocitrate dehydrogenase. Substrate availability is also an important factor in Krebs cycle regulation. The cycle requires a constant supply of acetyl-CoA, which is derived from the breakdown of carbohydrates, fats, and proteins. The availability of these substrates can affect the rate of the cycle. Next topic we will be learning with the help of next video. Thank you for listening.